afternoon, everybody, and thanks for joining us from wherever you are in the world. And this is the fourth in the series of our webinars, and we are glad to have Dr. Peter Imoesi here. So I'm just going to give a brief recap. I'm sure that a lot of us have read his bio, or at least we know something about him. But I'll just give a recap of who Dr. Peter Imoesi is, and so you can appreciate the level of information and the importance of why exactly we are here. So Dr. Peter Imoesi is a molecular neuroscientist and a research fellow at the Institute of the Medical Sciences, University of Aberdeen. And he also works, his research is for Taurex, I don't know how to pronounce that, but
Taurex.
Okay, Taurex Therapeutics, thank you. Both in the United Kingdom. He has a PhD in translational neuroscience from the School of Medicine, Medical Sciences and Nutrition, University of Aberdeen. He hails from Edo State, Nigeria. Could I please ask that everyone mutes their audio? Okay, let me just mute this. All right. So he hails from Edo State, Nigeria, where he acquired his first degree in biochemistry from Ambrose Ali University and completed his Master of Science degree in molecular biology with a distinction honors at the University of Hertfordshire. His MSc dissertation earned an outstanding grade for the overall best in project research at the University of Hertfordshire. Over the course of his career, he has won multiple research award grants and scholarship from the University of Aberdeen, Sir Richard Stapley Education Trust, MRC Discovery, Aberdeen Research Grants, and British Society for Neuroendocrinology, among others. So with various research achievements in biochemistry, molecular biology, bioinformatics, and even drug pharmacology, is widely sought after in the media, and he has been engaged in several media platforms, including channels CBC, NBC Africa, AIT, and the rest of them. So beyond the lab, he's married, and he has a blog. He has a blog, www.peters.blogspot.com. So you can go check that out after the webinar, where he writes about general societal issues, personal development, and motivate people for excellence. He's also a huge supporter of Arsenal Club. I have something to say about that, but I won't say it here.
Okay.
He has a published book to his name titled Absolute Success. And his favorite science jokes are, Miss Protein, can I catalyze your hydrolysis? And, oh, pepsin, don't be such a protease. Okay.
Biochemists will understand that better.
Okay, all right then. So let's welcome Dr. Peter Imoesi as he shares his presentation with us. Thank you.
Thank you, Kiki, for having me, and thank you for the opportunity to share a few of my thoughts as regards to COVID-19, where we are as a country and in the world at large. So let me see, I will try as much as possible to make this presentation really brief and straightforward. And I'm also aware that I've got a huge range of audience, so I will try my very best to ensure that the level of scientific terminology are brought down to the barest minimum. So let me see if I could share my screen. The host disable screen share. So you need to activate screen sharing as the host.
Sorry. I should have done that since. So it is now, you can share now.
Okay, so let me see. Okay, desktop one. Wow. So one circle. Let's okay. Okay, let me see.
Okay. We can see your screen. So
You can see my screen.
yeah,
So what part of my screen can you see?
the picture on your screen, the landscape, beautiful picture of the sea and the shores and.
Okay. So can you see this?
Yeah, we can see the slide now. Yeah.
Okay. All right. So we're going to be talking about testing strategy and journey to drug development. Here. Okay. Yeah. So basically, the coronavirus disease, like you know already, COVID-19 is from coronavirus disease 2019, and it's more like a single-stranded RNA virus. And sorry, I don't know what's going on. Okay, I need to click on the screen. However, we've got other known viruses before now that belongs to the coronavirus family. And the one that is more similar to the current virus we've got, which is COVID-19, that is caused by severe acute respiratory syndrome, coronavirus 2, share the same line of family with the Middle East respiratory syndrome, MERS, and the severe acute respiratory syndrome, coronavirus 1. You could call it 1 or just coronavirus. And they all belongs to the same class of family. So I would like you pay attention to the MERS and the SARS-CoV as we go forward. Now, the reason why I need to share this particular point again is to ensure that people should know that the current virus we have is not the first, despite its novel. We've got other known coronaviruses before now. We've got the human coronavirus, which is the NL63. You've got the human coronavirus itself, which is the HKU1. Then you've got the human coronavirus 229. Then you've got the other one, which is the human coronavirus OC43. So we know already that there are previous coronaviruses out there, but I'm going to share light on why the race for drug and vaccine may be slightly a little bit difficult. So my next slide, I'm going to explain to you the pathophysiology of COVID-19. Now, as you can see, I'm trying to take this. Oh, it's okay. As you can see here in this particular picture, I think the media have done justice to this over time. This is what a typical severe acute respiratory syndrome coronavirus 2 looks like. And from this spike here is the way in which it gained access into the host. And by host, I mean that human, for instance, through the lungs. And this is the gateway channel, which is called angiotensin converting enzyme 2. So with this, it gained entrance into the human host onto the cell. So basically, if we are to develop a drug or whatever we're trying to do, it means that we need to have a drug that could basically hit the entry points of this particular virus into the system. Now, this other cartoon I've got here was actually made by my friend from Canada. And you can see here that this is coronavirus itself. It goes in, then from here, it moves into the coronavirus enters into the cell via the ACE2 receptor I talked about initially. Then because it's a single-stranded RNA virus, what it does is it's kind of replicates using the host. So what that implies is, so you come into a house, for instance, that is not yours, but you're trying to take dominance of that particular apartment. So what you do is you look for similar characteristics of who you are in that particular building. Then you make a kind of pair, a pair, and from there you team up to destroy the entire flat or building. So that's what coronavirus does. It goes into the cell, and it looks for similar complementary genomic sequence, binds, then start replicating. And you can see here that coronavirus uses a host cell to create new virus. So that is a new human acquiring friends within the building, teaming up together to destroy the entire individual or the apartment of interest. Then again, the viral replication in the host continues. And this is when the virus has successfully taken charge of the host. You start seeing signs like this. You can see this young man here coughing, the virus is taking residence in that particular host. By the time he cough, he or she, if you don't do the right hygiene protocol, you cough into your hands, you're going to touch somewhere outside the environment. Then from there, you start transmitting or infecting areas where people who also come in contact with could also get infected with COVID-19. So this clearly explains why proper hygiene, hand sanitizers, and everything you need to do is highly important. Now, what is the behavioral pattern of COVID-19, and why is it that some country are secretly, I use the word secretly now, I'm not going to mention countries, but you be the judge yourself. Why are some countries secretly going through the herd immunity? Is herd immunity reliable? So to break this down clearly, herd immunity just simply means that you can see the cartoon here. The people who get infected, by default, they should build immune system. By the time they build immune system, so you build a defense mechanism. So you need little soldiers in your system that will help to fight subsequent infection. Now what happens is the weaker of this particular herd will then be protected. But is that particular mechanism reliable? Because COVID-19 is novel, we're not so informed of the behavioral pattern of the virus. And when it comes to antibody, how durable is this antibody when we're exposed to it? So that's why if you could see here, I said implication of herd immunity, reliable or unreliable. As we go further, I'm going to explain if that is reliable or not. Again, when you look at this other point here, sorry, I'm trying to move around my system. You have the implication of antibodies. So basically, IgM is a little soldier in your body that first comes into defense for a particular pathogen. So what IgM does is this. So if I have, let's say, cold, the first military apparatus that my system will release are called IgM. So IgM are going to fight and see if they could kind of defeat this particular pathogen. If they fail to do, they then recruit these guys that then come on board. So over time, these guys give way to this IgG. IgG are more the super guys. They come again to fight this virus. And if they succeed, IgG could, let me see. IgG could remain in circulation for a period of time. This example is measles. When you have measles, for instance, you could have antibodies for life in your system that subsequent infection, you're able to weather the storm. But for COVID-19, the peculiarity of COVID-19 is the fact that at what level of exposure do you need for IgM to remain in your system? So if you are mildly exposed, will it lead to more of your antibody present in your circulation? That is the first question. And that leads to why I said, is it reliable or unreliable? Now, if you make less of IgM, if you make less of these tiny soldiers here, it's going to lead to less amount of IgG, which gives you the real boost to fight against subsequent infection. In coronavirus, viruses, like I explained before, the first four coronaviruses, once you get in contact with the first four, within weeks, as a matter of fact, within 14, 15, or 21 days, your antibody goes out. The only ones that potentially stays behind is when you are exposed to MERS, which is the Middle East Respiratory Syndrome, and when you're also exposed to the first SARS, which is this. So this is SARS-2, it means there is SARS-1. Now for those two viruses, they are referred to as stubborn coronaviruses. They are really stubborn. And even as we speak, since the very first time they were reported, there is no vaccine, there is no known drug treatments or what have you. Basically, when I went on WHO website, the same recommendations we have for COVID-19 now is the same recommendation for the other viruses that have been around for the last 13 or 14 years. So basically, those two viruses, which is MERS and SARS, are referred to as troublesome viruses. For those guys, they could stay in the system when it comes to the antibody, they could stay in the system for 1 to 2 years. But for COVID-19, that is caused by SARS-CoV-2. We are not aware how long these particular antibodies will stay in your circulation to defend against subsequent reinfection. We're not aware at what level of exposure do you need to give you more of IgG, because usually with or without COVID-19, IgM will only last for a few weeks and they fade out. The only one that gives you the support you need in future fights is called the IgG. And at this point, we're not sure what to expect with COVID-19. And based on that, we can't really say, or we can't rely on herd immunity going forward. Again, when it comes to COVID-19, it changes every single second. Like by the next 24 hours, new information will just come out and you'll find a different story entirely. But for now, I've heard stories of people who are infected. They were tested positive with RT-qPCR. And when after 14 days of 10 days after, when they recovered completely, they tested them for antibody. There was nothing to show for it. It shows negative. Again, is that a function of the test kit, the rapid test kits, or what have you? That is still left unanswered. Now, when you look at this same antibody again, you could see this particular graph here is done by, I think this particular paper was published by some group of scientists in Oxford and Liverpool and across other universities in the UK. You could see that at day 0, this is infection. You could see IgM, which I said is a first defense response. This is your first soldier that comes on board. It peaks within 7 to 10 days. You could see at acute sample is really high. Then as the disease progresses, you can see the curve coming down slightly. And this is day 28 to 35. So it means that this is fading out. Again, this is a guide that we should need, that we require to fight every form of disease as far as COVID-19 is concerned, because if you're exposed, you should have high level of IgG. Now for this particular IgG, again, you could see that at day 0 up to the level of day 35 is not so peak. And what this implies is that it's fading out gradually. So what then will happen at day 40, 45, we're not sure exactly. So you can see clearly that this is actually fading out. So, and if this is fading out, relying on herd immunity or relying on test kits, rapid test kits, is not so efficient in the fight against COVID-19. Again, this same particular graph here is from the same set of scientists. You could see again at my far right, I don't know what it should be, your left maybe, you have the mild, you have the asymptomatic, you have the severe and critical cases. Now, if you take a look at the mild, you can see them, they are everywhere. So this particular graph is N of 40. So this is 40 samples. You can see again for the mild, you see individual patients, they are all scattered everywhere from day 0 up to day 60. Now again, when you look at, so this is for, on your left-hand side, this is for IgM, right? Now when you look up to day 60, people with severe cases, which is this particular color, more like green, they gradually disappeared completely when it comes to IgM exposure. When you look at IgG again, you can see that gradually it peaks, then it fades out again. So basically, the stories we're hearing about COVID-19 reinfection in China, or how reliable is the antibody in terms of fight against COVID-19, is here and there, and these are little proof to kind of consolidate what to expect going forward. Again, so far we've heard about drug repositioning or drug discovery for COVID-19 disease. This is a hot topic right now, and basically with my few knowledge into drug discovery and preclinical trials. So for Taurex, part of the preclinical trials, despite we have a drug already at clinical trial phase 3. And before I joined Taurex, basically, or before I started working for Taurex, this particular drug has been on for close to 2 decades, that's 20 years plus. So you could imagine how long it takes to develop a drug. Now, if we're trying to make a drug for COVID-19 disease, here are the things we need to look into. So if we intend to defeat COVID-19 using a mono treatment, then the drug should have the right efficacy to inhibit coronavirus infection life cycle. So what that means is that if you want to give just paracetamol for people infected with COVID-19, and that is all you want to administer, there are some parameters that drug should be able to do by itself. And because of that, because we do not have such drug at the moment, that's why most caregivers, they combine several drugs to treat COVID-19. So here is the bullet point for people who intend to do drug development for COVID-19. First, your drug should be able to block the viral entry. Remember the cartoon I showed you about the picture of COVID-19, the virus itself. And I said the virus goes into the host using the AC2 entry point. So basically, this particular drug, which is well known by everyone, chloroquine or hydroxychloroquine, actually has the efficacy to bind to that particular entry point. Remember I said that one drug alone is not enough to fight COVID-19. Now, if again, you want to use mono drug, that is mono treatment, the drug again should have the power to also stop the viral replication of SARS-CoV-2. And these are the drugs already being used in combination treatment. So what we have now is called combo treatment. So you're combining several drugs to treat one disease, and every drug playing its own role separately. Then again, if you intend to have one single drug for the treatment of COVID-19, the drug should have the ability to inhibit RNA synthesis. Like I said before, I told you that the virus goes into the system, it binds to the genomic materials, then replicates more of itself. Like I explained initially, that is where you come into a particular property, you make friends to destroy the entire property. So you need to have soldiers or a drug that will stop that particular process from occurring. And you know already, remdesivir is one of the drugs already being used, repositioned for COVID-19. Then finally, if you do want to have a mono drug, that's just one drug to do all the work you're looking for, that same drug should be able, again, to inhibit this particular receptor, the entry point in the cell, which is called the TMPRSS2. So this is where this same SARS-CoV-2 gains entry into the target cell. So you need a drug also that could inhibit this. So you can see all of these parameters that is required if you want to have mono treatment. So going for just one drug will really give a huge challenge to pharmaceutical industry or individuals who are more interested into drug discovery. So this clearly explains to you now why what we have currently is called combinational treatment. So this should give you a fair understanding why if the health givers are giving paracetamol, they are giving whatever they are giving to patients back home, this clearly explains, because COVID-19 will give you symptoms of high body temperature. And basically for you to bring that down, you need stuff like, let's say paracetamol, ibuprofen, and what have you to bring down your temperature. If you have cold, there are other drugs again. So it's more like combination of all drugs to treat just one disease. So imagine the number of tablets COVID-19 patients swallow on a daily basis. Going forward, how close are we when it comes to the vaccine itself? Remember I said before, if you go onto the World Health Organization website, we are told that there's no current vaccine for MERS, which is a Middle East Respiratory Syndrome disease. There's no there's no vaccine also to date for SARS-CoV-1, which is the initial severe acute respiratory coronavirus one we have before. Now going forward, where are we for COVID-19? If you can see clearly here, you could see that for this particular drug candidate, they are at preclinical trials. So this is really funny because this means that they are still in the lab testing on cell lines. They've not even gone up to, I don't know where they are anyway. Maybe they could be onto mice or rats at this stage, but preclinical trials is still really far away. And you could see the number of drugs, potential vaccine under preclinical trial phase, which are listed here. That's a lot. Now let's see if there's any other vaccine that has gone up to phase 1 or phase 2. So phase 1 basically is where you have the vaccine being tried on human, a small population, and you can see here you've got a few of them into phase 1, phase 2, phase 3, and 2 also. May I remind you that for the fact that this drug are up to phase 1 or phase 3 is not a yardstick that, oh, this vaccine are going to make it up to the final stage where it will be safe for human to be given to humans. Again, when it comes to vaccine, again, you will know fully well that the timeline, the quickest we've had, the very quickest we've had, is even Ebola, and that is 5 years for us to have Ebola vaccine. So you can imagine how long it takes to get vaccine out there for safe administration to the human population. Again, may I also remind you that when it comes to vaccine also, for coronaviruses, the thing about corona is this, and for most flu vaccine, here in the UK, most times we could go for flu shot every 12 months. That is because the antibody usually fade out. Then you need a new jab for after 12 months to get yourself prepared for winter. And that's simply because that this particular vaccine and antibodies you make, they kind of dwindle out along the line. Now, again, if you need a vaccine, if your coronavirus mutates to a different strain entirely, it means that you need to start developing another vaccine all over again for that new strain. So basically, if there is a small shift in your nucleotide sequence of a particular virus, that is a different organism entirely, just because of a few changes in the sequence. So with that, this is really a huge problem for the science community. And when NCDC say we should take responsibility, or when CDC say take responsibility, you know why we should take responsibility, and you know why testing is highly essential going forward. Now,